Welcome to today's webinar on how organic farming practices affect the soil microbiome. I'm Alice Formiga of eOrganic, and funding for today's webinar is being provided by the NIFA Organic Transitions Program. If after the webinar you'd like to learn more about the Soil Microbiome Project, you can visit the project website at eorganic.info/soil microbiome. Today, along with two researchers, we're lucky to have three farmer presenters. First, we'll hear from Elias Bloom of Cornell University, and then Jason Grauer and Jack Algier from the Stone Barn Center for Food and Agriculture. Then we'll hear from master's student Zoe Economos of Cornell University, and finally, Zaid Courtier of Norwich Meadows Farm in Norwich, New York. So now I'm going to hand things over to our first presenter, Elias Bloom.
Okay, and thank you everyone for coming. Today I'll be talking about cultivating a pest-suppressive soil microbiome on your farm. I'm going to start with a little bit of background about me. I grew up on a small-scale diversified organic farm outside of Omaha, Nebraska, and we grew many different small fruits and vegetables, from the potatoes you see here to summer squash. And we marketed through various different means. So here I am at a CSA that we hosted for a few years, and we also did farmers markets and high-end restaurants. And the farm was owned and operated by my mom, Rebecca Bloom, who you see here doing some seeding and who is now retired. I want to start, and I just wanted to say that much of my research comes from my background in organic agriculture. And so my growing up on the farm has inspired the last 10 years of research that I've been conducting, and the research that I've been doing for the last few years has been focused on the soil microbiome on organic farms. So I think it's useful to start by talking a little bit about the soil microbiome in terms of its functions, and I like to make an analogy with the human microbiome. So some fun facts about the human microbiome: that the human body is actually made up of more microbiome cells than actual human cells. And there are about 10,000 species of microbes that live inside of your body, everywhere from your airways to your mouth to your GI tract. And to extend this information a little further, we've come to realize that a healthy body is kind of driven by a healthy microbiome, and the microbiome in your body can help reduce things like gut inflammation. It helps to promote antioxidants and reduce your risk of infections. And so if we extend this analogy to the soil microbiome, what we know is that in a teaspoon of soil, it contains more than 1 billion individual microscopic cells and more than 10,000 different species of microbes. So in your entire body, you have 10,000 species, but in a teaspoon of soil, you have 10,000 different species of microbes. And some of the different functions of the soil microbiome include breaking down pesticides, improving soil aggregation, climate resilience, of course, nutrient cycling. And the two areas that we've been studying are pest resilience and plant defenses. So we think the soil microbiome can reduce pest numbers and increase plant defenses. And I'll talk a little bit more about that later. I think now that you understand the functions of the microbiome, I want to explain to you a little bit more about the diversity of organisms within the soil on your farm, and also some different ways to measure that diversity. And so we generally study two different groups, bacteria and fungi. And bacteria are tiny single-celled organisms that live in thin water films around the roots and soil particles. And they can grow rapidly. They can double in their population within 30 minutes. And they can also increase soil productivity and yields. And then soil fungi are generally larger than bacteria. They're multicellular. They prefer low disturbance. So bacteria prefer water. Fungi prefer low disturbance areas. And they're often key in decomposition of residues and the redistribution of nutrients. So we have bacteria and fungi, and then we also have different ways of measuring those bacteria and fungi. So we have what's called alpha diversity, which is just the number of species and how abundant they are. So here we have many different species represented by these shapes. And then, for example, the abundance of this shape here is 2. And so we have alpha diversity of your soil, which is just the number of species and their abundance. And we have beta diversity. And beta diversity is the changes in the species from farm to farm or over time. So if you walk to your neighbor's farm, you might say, oh, I have a different microbiome than you. And the way we measure that as researchers is through a measure called beta diversity. And finally, there's species identity, and this is really important. This is the taxonomic classification of a microbe. So what do we know about the genus or family or different type of nomenclature involved with that species? So now that you know a little bit about the microbiome in general and these different measures that we're using, I want to tell you a little bit more about our research. So what we're doing is searching for the pest-suppressive microbiome. And so what we think can potentially happen is that in more diverse microbiomes or microbiomes with particular species that we can classify using taxonomy, or potentially changes in the microbiome across farms, all of these different variables can potentially contribute to how a plant defends itself. And so what we've come to learn through research is that organic farms tend to have, for example, more diverse, more alpha diverse microbiome, and that this diversity helps plants create these chemical defenses. And these internal chemical defenses are produced by the plant to protect them from herbivores. So when you have a more diverse microbiome on an organic farm, your plants are better defended, and then you have more pest suppression. So what we don't know is specifically within organic farms, what practices are linked to microbiome conservation and the management of pests. And that's been the focus of our study over the last few years. So I'm going to introduce you to our study here. And I would like to just kind of tell you about how you would participate in our research as a farmer. So what we've been doing is working with farmers to have them submit soil cores. And so they go out to their field and they create a soil core like this, and then they mail it to us. And in addition to mailing us that soil core, what you would do is fill out a survey. And within this survey, we ask you about many of your different production practices. So how you're mulching your field, the type of tillage you're using, how you're irrigating your fields, all of these variables we think are potentially linked to the soil microbiome. And this project has been called the Organic Microbiome Project, and our tagline has been Donate Your Soil to Science. So once we receive your survey and your soil core, what we do is take the soil core and we run experiments, and we also conduct DNA extractions. And so in the experiments, what we do is extract that live soil microbiome from your soil core, and then we grow plants in it. And from those plants, we're able to measure the plant defenses, and also we're able to put insects on those plants and see how many progeny or how many baby insects they produce. And so from that, we can get an idea of what types of soils are changing plant defenses and their capacity to suppress pest populations in a laboratory environment. And then with the DNA extractions, we're able to look at that alpha diversity, the beta diversity, and also the species identity. And so we're able to say, what types of different microbes do you have in your soil? And then how is that related to plant defenses and pest suppression? And then we can also link all of that using the survey tool to your farming practices. So I'll talk a little bit about the amount of participation that we've had so far. So here is a map of New York. We're up here. And anything in red and blue are areas that we did not receive samples from, but every other color throughout the state are counties that we've received soil samples from for our study. So we've worked with about 85 farmers so far. And this ranges from farms on Long Island, like the one you see here, where there's houses in the backyards of the farms. It's a very urban area, to farms in upstate New York, which are very rural. And so you can see we've had a wide suite of participation from many different farms, and they come from many different regions of the state, and they all use many different types of practices. So from all of those submissions, we're able to start filling in this picture of the organic microbiome. So far we've identified 9,343 different types of bacteria that are living in the soil on organic farms. And this is the range here. So about 1,000 to 6,000 different types of bacteria on your average farm. So if you were to go out and take a teaspoon of your soil, you would probably have between 1,000 to 6,000 different types of taxa or species of bacteria. For fungi, we found an additional 6,740 taxa. So those are like species, but we're not able to always identify them all the way to species. And that ranged from about 650 to 1,600. So again, if you go out to your farm, you might find 650 to about 1,600. So in total, on organic farms, we found more than 16,000 different microorganisms from the groups of fungi and bacteria. So this is actually more than the 10,000 that I told you about earlier. And then from there, we've identified an additional 114 taxa that are associated with specific farming practices. So actually a very small number of these were able to relate directly to farming practices. And then for beta diversity, what we found is that if you were to go to your neighbor's farm, you would actually have about a 20 to 30% change in the microbiome between your farm and your neighbor or someone else in our study. So this is, I think, the table that many of you are most interested in finding out about. So what are the practices that you can use as a farmer to promote the microbiome, plant defenses, and pest suppression? So for this first column here, what we found is about 25% of the fields in our study use soil pesticides that are allowed in organic production. And what this appears to be doing is increasing the abundance of specific bacterial taxa, so those species identity effects. And so when these bacterial taxa increase, they reduce plant defenses. So what we're finding is the use of soil pesticides tends to have a negative relationship with plant defenses. So when you use a soil pesticide, plants are less able to defend themselves. And then downstream from there, we've found that it can also lead to increased abundance of pests. So the next one is actually kind of interesting, and we've kind of been going back and forth about what to tell you today. But about 55% of the fields on the farms that we've studied use compost applications as a nutrient input. And what we found is that some compost applications seem to be increasing the beta diversity of fungi, so changing the kinds of fungi that are found on farms. And so with these increases in fungal beta diversity, we've actually seen some decreases in plant defenses. And so what we're going to tell you today is that composting or compost applications could have a negative or positive impact on your microbiome. And it seems to be dependent potentially on either the compost that's being applied or the microbiome that's already present on your farm. So now on to the positive. What we found is that drip irrigation is being used in about 56% of the fields that we studied. This tends to decrease bacterial beta diversity. So it actually, what that means is that it makes the bacterial species more similar on farms. And so with these decreases in bacterial beta diversity, we found that this actually helps to increase plant defenses. And so you'll remember from the early slides that bacteria love water. And so that's exactly what we found here. So apparently what we found through our study is that drip irrigation has a positive impact on plant defenses, and that can then in turn reduce pest populations. And then the final area here that I wanted to talk about is no tillage. What we found is that the farms we're working with are using some form of no tillage, about 42% of them. What this seems to do is reduce some common fungal taxa and increase some specific bacterial taxa. So what we're seeing is changes in the alpha diversity of fungi, but we're also seeing changes in the species identity of different fungal and bacterial species. And these changes in specific fungi and bacteria are associated with increases in plant defenses. So no tillage seems to have a positive relationship with plant defenses. This, I think, makes sense and is well supported by the literature. And it has negative in it and also helps to reduce pest populations. So I'm just going to close up with a few thoughts here. It seems that no tillage helps to support pest suppression through the microbiome. And I know that there's a lot of problems with scaling up no tillage, so consider when and where adoption is possible, even at small scales on your farm. The next area is irrigation. Again, irrigation was highly linked to bacterial beta diversity. It appears drip irrigation promotes pest suppression via the microbiome, but you're going to have to weigh whether or not the adoption of the adoption of drip irrigation is right for you. For pesticides, it appears that pesticides are changing the identity of microbes on organic farms, and it has negative impacts on plant defenses. So finding ways to reduce or eliminate the use of soil pesticides is important. And then finally, for composts, it appears that composts interact with the farm microbiome in unique ways. And so I recommend monitoring your applications closely. And we'd love to hear what your observations are in terms of if it's increasing or decreasing pest pressures on your farm. And we think it potentially could be working through a nutrient cycling mechanism. So I'd love to go through all the thanks that I have for everyone, but big thanks to Claire Castile, who's the project PI, and a project mentor, Shadi Atala, who's helped a lot with this study. And I'll pass it off because I know I'm over time.
Jason Grauer. I'm one of the senior directors at the Stone Barn Center for Food and Agriculture in Westchester, New York, down in the lower Hudson Valley. To share a little bit about our work, I'm going to give you kind of a brief overview of the center and the work that we're doing as a farm education and research campus. And then I'm going to pass it over to Jack Algier, who started this farm that we're working on and has created and strategized over the last 20 years the farming systems that we're using on the farm. And our organization is a fairly unique organization because we really attack food culture issues. We're attacking the idea of regional agriculture and regional food shed from both the supply and the demand side. We're a farm, but we work in close partnership with a restaurant that is supporting farms throughout the Hudson Valley and throughout New York State and the region in the Northeast. And all together, collectively, whether we're looking at food from the plate or from the kitchen, or whether we're looking at it from the agricultural or ecological side, we're actually really just focused on the soil. So soil is the heart of this organization and what we're promoting, soil health. And a deep understanding of that soil health is what we're trying to accomplish. So here's just a beautiful view of actually the State Park Preserve, the Rockefeller State Park Preserve, who's one of our other long-term partners at the Stone Barn Center. What we think is important to share is that we have both, we are running farming systems on private land that the nonprofit owns, but also the majority of the land that we're working on is in partnership with a publicly leased land. So we have a 50-year lease and an arrangement to collectively manage these landscapes and to use agriculture to do so. So reducing the need for any kind of chemical interaction and reduce the amount of machinery used, and instead inviting in mixed, diverse agricultural systems to care for the trails, the forests, and the open meadows and pasture lands that we have. So, and I was just going to say, here are the 4 categories in which we're organizing what we're doing at the Stone Barn Center. So at the start, agroecological research and methodology. So this is what I was referencing, and Jack's going to speak more about the details of how we think about soil, microbiology, and the implementation of this. But at the core, what we're doing is running a farm that's focusing on research and focusing on systems. So on all these different landscapes that we're managing, which is upwards of 500 acres, we have several different agricultural systems rotations that we're running. Many of them are integrated animal and vegetable and pasture, and some of them are very specific, like our greenhouse system or our main vegetable system that runs kind of like a traditional organic crop rotation. As a nonprofit, we're doing this to also share the findings and to share the food and to share this experience that we can create on this farm with the community. So community engagement and education is another core to what Stone Barns is doing. We do this internally and we do this externally. So on average, we're running tens of thousands of tours and lunch trays and dinners that people in the community are coming up for and engaging with annually. And additionally, we're running certain programs where we go offsite into our community and into areas that are well underserved, and we engage with those communities, sharing the ideas, the research, and the findings that we have, but also in a bi-directional way to also work with the communities to evolve our educational components to fit and to learn from these communities as well. So one of the programs that we're leading right now is called LEAF, or Leading an Ecological and Accessible Food System. And so we're working with two communities, or two aggregates of communities, one in New York City and one here in Sleepy Hollow in Westchester, to basically provide a CSA of plants to grow, food to eat, and engaged learning to exchange ideas and to share and to use the expertise of farmers for the growing, cooks or chefs for the cooking, and then also just the community sharing ideas and sharing their experiences to see if we can improve our relationships as a whole to a regional food system. Next, we move on to partnerships and advisory services. So I mentioned two earlier, but there are two very strategic long-term partnerships, one with the State Park Preserve in New York State, and the other with Blue Hill, a restaurant partner and a partner of Stone Barns since the beginning 20 years ago. We're also expanding this to work in strategic partnership with other food and farming organizations in the future. And one of the big key drivers of sharing this information and working together is the advisory services led by Jack, which is essentially consulting and helping farms and organizations that want to implement similar systems to Stone Barns on their property. Lastly, I would touch on food innovation. That's something that we're very known for and something that we feel very uniquely positioned to focus on. And this is the intersection of agriculture and food, you could say. And so this is where everything that we do, we don't just see how it works ecologically, how it works in a farm system. It's not just about the yield. It's also about understanding how that production can influence a food culture. So we bring it into the kitchen, we evaluate these things together. So we run hundreds of variety trials with a variety of plant breeders, whether they're from public universities or whether they're from private regional seed companies. Anyway, we're looking at kind of identifying and understanding food from that holistic approach. So here's just a little bit of an example of what I was describing. Our farm team is broken up into these four main teams, just to give you a little bit of color here, and then we can move through some of the other slides a little quicker. But here our teams break up into livestock, crops, which is vegetables, flowers, herbs, and grains, and ecosystems management. So you can see the compost windrow here, and also landscapes and orchards and perennial areas. And then lastly, the ecological monitoring. So this is something that I think is fairly unique to only certain organizations. We are fortunate enough to have a team that's monitoring all these farming systems and is able to engage with and partner with outside researchers, like in this project, to be able to make sure we're tracking data properly and we're executing any kind of data collection and analysis to the highest degree. Here's just an example of what we were talking about earlier as far as public tours, membership, dining, lots of visitors coming to our site, and we also getting off to engage outside of our property. Okay, just to touch on some of the research projects that we're doing with this collaborative of people, and I'm going to pass this over to Jack. So within these systems, we have a number of ongoing research projects, some multi-year, some literally ongoing. In the farm systems, we recently received a SARE grant to study one of our cropping systems, the lay rotation, which integrates all those farm teams mentioned before. And so we're exploring through studying the soil, the impact of this rotation on the soil health and the carbon over time. We're working with Washington State University in a research project, both in Washington and in New York, to evaluate the impact of buckwheat on production systems. So we're seeing where if buckwheat is entered into a vegetable rotation, how does it affect soil health? How does it affect weeds? And how does it affect production? The Conservation Action Plan, which I will be spoken about a little bit later on, is an ongoing project, an ongoing research project where we're doing water quality, soil health, and biodiversity monitoring to analyze the impact of all the farm systems and research systems on the landscape. Within crops, like I mentioned, we are doing seed breeding and seed saving and hundreds of variety trials annually and sharing that information out with farmers. On the livestock side, we're working on very unique projects. One, to revitalize the dairy industry in the Northeast, helping from an economic standpoint, and the other is to really explore and deep dive into how waste feeding can be a viable option for farmers to be utilizing resources in their region and reduce the costs and purchases of grain on farm. And I'm going to turn it over to Jack to speak a little bit more to the holistic systems, soil, and the microbiology, and how we think about all that within these programs.
Okay, great. Thank you, guys. In the interest of time, I just want to make sure we're good. Do a time check. It's 2:30. I'll be brief here, but Jason did ask that I speak to some of these things, and I think it's valuable for this group here. And there's many people on this webinar who have been great inspirations. Just the idea that soil health is a term that is universally recognized now in the academic world is a huge step. And some of that is, I would thank Harold, who's on this call, for some of that, and a lot of other soil scientists who have made a huge dent in this understanding of ecosystems as living systems and not just an assembly of parts. So I'm going to talk about regenerative, organic, and agroecology. Those can all be together as a word or 3 separate words here. But what's important is that we're looking at these things slightly differently. And we look at our system in general through the lens of agroecology. And that is that we have a scientific discipline, an agricultural methodology, and a social movement happening kind of in tandem with each other. And that's how we look at when we're bringing in hundreds of thousands of people to our site. Unlike most farms, we are very public, and there are people here all the time who've never been around farms or around animals. And it's really valuable for us to be able to demonstrate working living systems, but also makes our work a little bit different than most. We don't have the sort of seclusion to do certain kind of agricultural practices, so we need to really show forward-facing and innovative practices all the time to have a good narrative. Generally, health, ecology, fairness, care, these are terms that IFOAM, for example, uses. You can scroll ahead. And that's a picture of our landscape in general. With soil health, I've always kind of considered, since we're looking at the soil as a living being, one of the ways that we look at the system is for the sake of fitness. And there are kind of 3 things that we look to do all the time in balance, is to make sure that there's a lot of movement and activity, cultivation and crop diversity and rotational succession, combined with good moments of uninterrupted rest and dormancy, perennial systems and fallow, and also just to have good metabolism, mineral, cover crop, this kind of diet that happens in the system. And these 3 pieces are really instrumental in a long-term rotation. And I think it's good to keep it very general like this, because everybody has different crop selections and all this. But for the most part, this is, from my view, something that's kind of become a denominator for the way we look at all of the systems, whether they're pasture, large landscape, or greenhouse. So continue. And in our greenhouse, we have a half-acre range in a 10-year rotation, year-round cold system. And we really rely on the health of the soil in that space. And actually, Elias made the point about compost being sometimes helpful. And I think one of the big parts here is that rotation and combination, and even the recipe of the compost itself has a very significant impact on this. So we have over time decreased the amount of compost, and that's partially because the soil system we have has matured and requires less. But also we've increased cover crop systems even inside these soil houses, which is a little less active than we would use outside. But nonetheless, we're using our indoor systems as cold systems so that we can break pathogen cycles. This is a 20-year indoor greenhouse system that has had very little, if any, organic treatments or anything like that, just composts and kelps and materials that we put into these soils. Next. But there was one of the things about us being this innovative space and having great support, the kind of privilege of having a community of support around us and chefs and members that are encouraging us to take risks and try new things. I have, for a long time, questioned this idea of sustainability in an annual vegetable cycle. And I've been curious about what old systems had been in place and how we could find new modern approaches to them. And we were also in the position of restoring hundreds of acres of pasture land in the park preserve around us, the state park preserve. So we started to trial pilot lay rotation cycles, which are long-term, or 5 or 6 year perennial pasture, and then a series of cover crops and grain and legumes and vegetable crops that basically exercise the soil and allow us to condition the space and reapply rich, diverse forages for continued pasture. But we've also learned that that actually is a very valuable process for restoring native habitat, which we've gone to 50 acres of restoration of native warm season grasses on a hill using these same lay rotation cycles, pulling grain, cover crops, that sort of thing to get that work. I think it's a really interesting area to study because our soils in particular, which are all very much sandy loam, have very low aggregate stability to them, and they actually like to have longer term perennial cycles. And so the vegetables and grains actually benefit greatly and require very little fertility addition in these longer term animal cycle pastures to revigorate the soil. So it's turned out to be quite a sustainable cycle. It does require a lot of interaction that most of our agricultural communities don't have the capacity to, which is to have the grain growers work with the crop growers, work with the livestock farmers, and it's a lot of coordination. And in many ways, Jason and I spend most of our time wrangling that. So this is a thing that we're experimenting with. It's working really beautifully. It's also very complicated. And just the thing we wanted to share with you because it is absolutely working and giving us a lot of hope. Next. The last 2 slides here are just to show that the agro-ecosystem is really a research platform, and we're working with more partners and looking at how, in our particular area, Pecanico Hills here, we are essentially in charge of our watershed from the Buttermilk Ridge down to the Hudson River. So we're able to track and monitor all kinds of things from water and carbon to biodiversity and habitat. And that's been a really interesting project for us ongoing with a lot of academic support and partnering from Cornell to Columbia to Pace and Yale. And then lastly, just to mention that these stakeholder models around climate research are kind of up and above the ecosystem itself, is that our region has huge capacity in the future, just something really bright. All of us are in New York and in the Northeast here. This is a very bright spot, very bright region for the future as climate change continues to desertify places and make it more difficult for more stable growing environments. The Northeast has a fairly positive future considering. So we're studying this movement, this kind of harbinger for what's to come. We're doing a lot of research on warm season grass restoration and climate adapted crops as the USDA zones move north and weather patterns change for us. So we're really happy to have partners in that way. they really help us go beyond the kind of practitioners that we are. I think that's it. I'm going to leave it there, and thanks for the time.
My name is Zoe Economos. I am a master's candidate in the Castile Lab as part of Cornell University, and I'm here to talk about the content of my thesis research, which is using cover crops to enhance dry bean stress resilience. So to introduce myself very briefly, I started my academic career in 2018 at my local community college, and I wanted to be a farmer. So I went into the Sustainable Farming and Food Systems program there. But throughout getting my associates, I realized that I wanted to have kind of a different role, bridging the gap between researchers and farmers and encouraging information transfer that way. So currently, I am working on generating information that helps farmers make decisions, and just helps them have more tools in the toolkit, as climate change progresses and stresses get more prevalent. So I made a little graphic to sort of illustrate the research that I'm currently working on, kind of outlining what I see as the final process that I see my research supporting. So farmers, like all of you, are constantly making decisions on their fields based off of the external stresses and stimuli that their fields are facing. And this is especially relevant in climate change. So we have increased temperature and increased drought. This farmer also has on the left side aphid infestation on one part of their fields. On the other side, they have chewing herbivores. And so they're considering what to do. They're wondering about irrigation, maybe for more environmental factors. They're wondering about pesticide applications. And they're also considering more organically oriented management options like cover cropping. But within those organic management options, within cover cropping, there's also more nuance. So which cover crops perform best with which systems. And throughout all of this, I'm also interested in the soil microbiome, which is at the bottom here. So trying to understand how the soil microbiome mediates these effects and how differences in the soil microbiome can translate into different pest defense applications. So I want to give you a little bit of an overview of cover crops and how they can be used to suppress pests and pathogens. I'm sure a lot of this is, just review, but I want to, just state some of the literature. So one study found that white mold and leafhopper populations were reduced in dry bean when grown with cereal rye cover cropping in comparison to tilled controls. And another study found that both canola and cereal rye cover crops increased soybean yield with a soybean cyst nematode in comparison to a tilled control. But there's nuance. I think everybody here also acknowledges that when you translate research into life systems, there is going to be complexity. So one study found that fall armyworms actually perform better on corn when it's grown with radish cover cropping and worse with triticale. So triticale performs a more positive functioning for corn with fall armyworm herbivory. And then additionally, another study shows that cereal rye cover crop can increase lady beetle populations, but also can increase aphid and leafhopper populations in snap bean when compared to a tilled control. And so I'm really interested in questioning why these different matches and mismatches of cash crops and cover crops matter in terms of pest defense, and also sort of translate that into different pests. So I want to sort of fundamentally lay out why I want to consider dry beans in my study systems primarily. I think a lot of people here are very familiar with dry beans and legumes in general. I know that Jack mentioned a little bit about legume cover cropping and legume crop rotations. They are the most important legumes in human food systems. And as many of you probably also know, they can fix nitrogen, which is an insanely beneficial aspect to cropping systems. And the consumer demand for dry bean production has been up 60% since 1990. So it has steadily increased, and research on how to have resilient and reliable, sustainable dry bean production is indicated there. So I want to give you sort of an overview of the setup of our field trials, where we sort of combined a bunch of different cover crop treatments with a bunch of different cash crop treatments. I'm going to be primarily focusing on dry bean, as well as canola and cereal rye cover cropping. I'll walk you through it. But at first, we were conducting these field trials in both, I'll show you in the upper right here, there's a map of New York with the locations that we did this trialing in. The yellow star is Musgrave Research Farm in Aurora, New York, and the red star is Hudson Valley Farm Hub in Hurley, New York. And so we have essentially these like replicated block designs. So each of these blocks was repeated 4 times in each of these locations. And it essentially trialed all of these different cash crops you see on the vertical axis of this block here. You have sunflower, soybean, dry bean, and sweet corn. And then up on the horizontal axis, you have a tilled control, canola, hairy vetch, cereal rye, and then a hairy vetch and cereal rye combination. I'm primarily interested in dry beans, so I'll walk you through like an example of what we would do in one plot within one of these blocks for a dry bean crop grown with cereal rye. So essentially, researchers would go out to each plot within each of these blocks, look at all of the dry beans grown with cereal rye. The cereal rye had been grown in the winter season and then roller crimped, and then the dry beans had been planted. And then at 10, 20, 30, and 40 days after planting, the researchers came out to the field, assessed it, said, okay, how much of this plot has just generally herbivory damage. And then they generated an interval rating from 1 to 4. So 1 being no damage really whatsoever, and 4 being, oh my God, crazy amount of damage. And so over those 4 different time points, interval ratings were generated and then averaged. And so I'll kind of show you later the results for canola and cereal rye cover cropping treatments grown with dry bean. But I do want you to know that we did these trials with multiple different cover crops and multiple different cash crops. And there's a lot of data that I can't show you today because it would take a long time. But we also did lab experiments. So in the following years after we did the field trials, I went ahead and I went out to a different cover cropping trial in Caldwell Field at Cornell as well. And I sampled soil grown with canola fields and cereal rye fields. And so it was from the live growth of the cover crops. And from both of these soil samples that I did, I extracted the microbiomes. So this is a method that I believe Eli was also using. Essentially, you take a little subset of the soil sample, you put it into these flasks that you see here with these like brightly colored orange caps. And then you add a nutrient solution that feeds all of the different microbes that are there, and you shake it up, and then you have a slurry that you can apply. And so what we typically do is we start applying the slurries at the start of planting in the lab. So we'll grow dry beans with these soil slurries, and we'll continue applying them so that we make sure that we have a constant composition of the microbes there. And then from those dry beans that are grown with these separate cover crop treatments, these cover crop microbiome treatments, and additionally, I also did a tilled soil sampling, and I extracted microbiomes from there to compare. We looked at beet armyworm herbivory. So beet armyworm is a really prevalent chewing herbivore. I'm sure some of you have come into contact with at least either beet armyworms or armyworms in general. So we went ahead, and after all of those dry beans had been grown in the microbiomes for about 3 weeks, we took a leaf from each of the plants and we put it in agar, and then immediately we put a beet armyworm on it, and we let them sit there for 24 hours, and then we measured how much of the surface area of the leaf had been eaten at the end of that time. And then additionally, we also did cowpea aphid survival trials. So cowpea aphids, different from beet armyworms in that beet armyworms are chewing insects, and cowpea aphids are more piercing, sucking insects. So they are much more subtle in their herbivory. So we essentially what we did is we had these tent cage setups, and it was the same thing. Dry beans were grown with these microbiome slurries and these different cover cropping treatments and a tilled control. And then we put clip cages onto each of the leaves on each of the plants, well, on one leaf for each of the plants. And we added cowpea aphids to those cages. And then after 24 hours, we removed the adults. They had babies. And then after 5 days, we measured how many of them had survived. So we have some results for you. From the field trials that were conducted that I mentioned with the block designs, looking at those pest ratings, we found that in dry bean, pest damage was actually significantly reduced in the plots that had cereal rye and canola cover cropping. So along the vertical axis there, you see it's the average pest damage in the dry bean plots. And then on the horizontal axis, you see all of the different cover crop treatments. And you see that canola and cereal rye have significantly less averaged pest damage in comparison to both the tilled control and the other cover crop treatments. So interesting start. But if we go even further with our microbiome transfers, we see that the microbiomes generated from these cover cropping treatments actually have differing impacts on different types of pests. So we see that the cereal rye soil slurries actually reduced beet armyworm herbivory, but increased aphid survival. And that's the most important take home, but I also want to really highlight. So I'm going to walk you through the graphs themselves. So on the left-hand side, we have the results of the aphid survival test. So like I mentioned, just a percentage of the babies that the aphids popped out that actually survived after 5 days of being on these plants that were grown with these microbiome treatments. And we found that cereal rye actually had incredibly high aphid survival percentages in relation to the canola cover cropping microbiome treatments. And none of them were significantly different from the tilled control, but they were different from each other. And you can visually see it's pretty striking. And then on the right-hand side, we have the beet armyworm or the chewing herbivory assay. So you see here, it's basically like the opposite trend, which is really exciting to me. So we see that the canola actually has a much higher percentage of beet armyworm herbivory in comparison to the cereal rye. And cereal rye has significantly lower herbivory compared to the tilled control. And it's like, if you put these graphs next to each other, that's just like, wow, awesome. So like, what's happening? And what does this mean for farmers and for researchers? So I want to talk a little bit about take-homes and next directions. So when I think about how to distill the results down into like, okay, what can be done in the field, or what can we start trialing in the field or in your own plots? I would say if you're growing dry bean and your field has aphids and you're interested in using cover cropping as part of your toolkit in pest suppression, you might consider canola instead of cereal rye. Whereas if you have chewing herbivores in your field, like beet armyworms, for example, you might consider cereal rye cover cropping. So it's a really interesting like comparison here that I think deserves more research and also deserves more trialing. Not saying that it's something that can like fix everything. I think everyone here is aware that, you know, you need an arsenal of tools to combat any of your pests and pathogens on your farm, but it is an interesting piece of the toolkit as far as I'm concerned. And then in terms of next steps, so this is just a piece of the puzzle, I think. As I mentioned, I believe I mentioned that the cover crops in the field were roller crimped before the cash crops were planted. But I think that that brings up an interesting question, because roller crimping isn't necessarily done by everybody. I mean, for example, some people flail mow their cover crops into their soil. So what kind of cover crop termination strategy gives the best outcome? That's the question. Another question is also talking about like cycling, like summer cover cropping versus fall cover cropping. I didn't put that on the slide, but that's another really interesting question that I thought of when I was listening to Jack talk. And then also thinking about different cover crops, cash crops, but also locations, because from this research, it's sort of apparent that there are very specific interactions happening between the microbiome, cover crops, and the associated cash crops. So that really says we need to do a bunch more trialing. And finally, we want to talk about microbiome sequencing. So the microbiome transfers that I did, it's basically just these microbiomes are different, but we don't necessarily know how. So I think sequencing the microbiomes to figure out what specific microbes are present in the different cover crop treatments is very indicated because that could potentially inform further innovations. So I went a little quicker. So I hope that everything was easy and clear to understand, but I'm hoping that I can answer some questions from folks. And I just want to thank everybody for coming and listening to everyone talk. I think this is an amazing opportunity. And I also want to thank the folks from the Castile and the Ryan labs who really helped me with my research. So Dr. Claire Castile is a prominent one, Dr. Elias Bloom, who's also here today, Dr. Uriel Monalid, Dr. Chad Narons, Ethan Eddy, Shoyamjeet Ray, Tyson Murad, and Dr. Matthew Ryan, and many more for discussion and inspiration. Thank you so much.
So I'll talk a little bit about myself, my background, just for a minute so people get some context of where I come from and what has influenced what I do. So prior to me starting the farm in 1998, I had a 7-year stint with Cooperative Extension in a Cornell Cooperative Extension with a dairy livestock team. And prior to that, I was working on a PhD, which I never finished, and in Kansas. And prior to that, I spent 7, 8 years between, a little more than that, between Wyoming for college, as well as worked for Farmers Home Administration. So, and then I got bit by the farming bug while I was working for Cornell, and the rest is history, and it's been about 27, 28 years. So, literally started in my backyard in 1998, and currently we have added another 50 acres about 60 miles north of us. Rapid expansion has been basically our what we've been doing all this time, and it has been very exciting in the sense that as Jack and Jason have talked about in much more detail than I'll be able to, the research part, although we don't get to do it at the depth that they do, we try to apply the results to make sure that we have a good farm and a profitable farm. So we are now more like 300 acres, and 200 by the end of this season will be around 240 high tunnels covering about 22 acres. And our business has evolved radically from the beginning, from basically farmers markets to a very heavy CSA presence. We had over 2,000 and about 3,000 shares at the peak in around 2011. And then the CSA movement kind of changed. And so now we're more like 50% restaurants and 50% retail, which has given rise, the restaurant side of things has given rise to the thoughts of, more about community. And given that chefs can be multipliers, we have spent a lot of time working with them and trying to figure out good food going to not just the 3 Michelin star customers, but the same good food being accessed by everybody. And so we participate, for example, in youth farmers markets where the goal is to get youth involved in agriculture and also talk about things like nutrition and good food. If we can advance the slide, please. Thank you. What we have done more out of our love for eating good food is we continually add variety to what we do. So 1,500 is about probably a little more. Every year I say I'm going to cut back. It doesn't really work. I wind up adding more and more varieties just because it's a fun activity, and it makes for great marketing as well. High tunnel production began for us. About in 2003 started to travel to Egypt during the winter, and I began recruiting my farm workers from there because they had worked on organic farms and they had a lot of extensive experience in high tunnels. And so that's what kind of fueled our rapid expansion into high tunnels, and our reliance on it. We're in a relatively cold spot. When I first moved here, we were a 4B, and now we're a 5B. So it's a little bit scary from that sense, but we had 49 degrees 2 nights ago, so we still are in a relatively cold pocket. So the high tunnels have really changed what we do. And after 20 some years of having high tunnels, we also learned that what happens in the high tunnel is radically different than what happens outside. And so there's a lot going on any given day, and trying to bring science into what we do is rather difficult, given that there's a bit of a language barrier. I mean, I speak Arabic very well, but there's also almost a cultural barrier, in the sense that we are organic because we believe in it, and most of my farm workers are inorganic because it's basically offers more money and more opportunity. And so that's an ongoing, it's ongoing for us, training our people on what does organic really mean, and now the new buzzword regenerative, and how do we maintain our soils, given the fact that one of our perennial problems, and it still is a problem, it's less of a problem now, is that land access is very difficult in and around us. The best of lands is mostly taken up by dairy farms. It's hard to find the proper land, or the best land for growing vegetables. So we have to make do sometimes, and that means, you really have to understand your soils and work them well and keep them in condition. And so that's what we have tried to do and continue to do, and it's an ongoing process, and it will never end. As university collaborations and grants, though we've only been doing that now in earnest for around 3 years, since I've been able to work with somebody who I actually met while I was working for Cornell, and more or less hired him on to help us with that. And so we have a multitude now of things going on. I'll talk about that a little bit more in another slide. Hospitality and relationship is at the core of our business. That's like key to everything we do. So I spend most of my time in New York City during the summer months, interacting with my customers and dreaming up new ventures. And it's the difference between who we are now and an average farm, I would say. We value this. We're truly about hospitality. It's not a fake thing that we try to use to get people to buy from us. It's just who we are, and it has served us well. And I would encourage people to a lot of times I see farmers that aren't doing well, and a lot of times it's because their skills with human beings is not that great. Just some if we can advance the slides, just a little bit of the types of things that we grow. Jicama on the left. Jicama is really a warm weather crop, but it has worked okay for us. And the beans there is actually from I don't know, about 8, 9, 10 years ago. Dan Barber from the restaurant that was mentioned earlier, Blue Hill, had a meeting at Stone Barns about breeding, and I sort of invited myself, and on the back of an envelope, Dr. Mazurik and I and John Hart started a bean breeding program, and those are some of the iterations of beans that we worked on. And right now there's about 11 varieties that are out there that are potentially going to become commercial varieties on the open market. So that's, again, that's the fun part of what we do. And as you can see, we try to grow just about everything. I'm working on mangoes. By the way, Jack and Jason, you owe me a couple of bananas. It's, it would be nice, but if we're ever able to grow mangoes, I think it might be a very disturbing time in our neighborhood. What are some of the things that we're working on in terms of grants and collaborations, ongoing collaborations with Michael Mazurek, Dr. Mazurek, and other breeders across the country. But we have a very close relationship with Dr. Mazurek, given the proximity and given that he is a friend. Row 7, that evolved from our association and our beginnings back with Dan Barber about 10 years ago, and that continues, and we trial things for them, and we also grow some crops for them now under their new model under row 7. This year we have Dr. Mazurek, along with some other universities, is doing the International Pepper Conference. So we are growing a bunch of varieties of peppers specifically for people to come and see as part of that event. Grants that we're working on, they're about to end. The VAPG, about 2 years ago, a little over 2 years ago, we got a producer value added grant, and we took a little bit different approach, and that will be talked about in the next slide a little bit, and that is that how do we add value to our product by promoting, for example, the fact that we are working and striving towards having very good soil and soil health. And can we communicate that and monetize that? So that is, how do we monetize some of the attributes of the services that farmers can do? And so this is if we want to move on to the next slide. So part of that is our work with Joseph and Dr. Van Ness on this. This is the culmination of that. This particular graphic shows, it's a scorecard of how we're doing against others. And that also is combined with a piece of software that we've been working on forever, which is now it's Fellow Farmer. Now it's called Holo, where a complete software program that will allow you to plan crops, market crops, keep track of all of your data as related to crops, including some form of AI where you can enter variables that allow you to look at growing degree days and any other variables that will allow you to better manage what you're doing is also part of this. So this particular graphic, it's everybody does soil tests, and therefore this is a way to basically for us to show off how we are doing. It's a report card, and hopefully we can also in the future monetize it. We are going to use it as part of our website that we're constantly changing to allow us to sell products with attributes, and this is definitely one of them. The process of improving soils, if you want to move on to the next slide, please. I think I want to talk about it a bit more from how our farm has evolved. I started with basically quit Cornell and started a farm and did not have a ton of money, and I just needed to do that. And so, you start off with a few acres, and demand was so high that, a perfect cover cropping system that I always envisioned never materialized. It has only been the last 5 years where I've been able to say take out between 50 and 80 acres and at least put a fall cover crop on them. This year is the first year that we've actually are doing a multi-year cover cropping sequence because I have the acres. It took a long time to be able to, one, have the profits or enough profits to be able to take a bunch of land out of production and not worry about the future. So that's sort of, the fact that, we want to be a farm that pays our people well, pay ourselves well, have enough money so if a catastrophe happens, we're not dead in the water. We don't borrow from banks. And so, basically existing on cash flow. We're relatively now large, so it's a lot harder to do, but we've so far been able to do that. That has basically affected how we can, what we can do in terms of, cover cropping properly and having enough acres to take land out of production for, say, I would love to do it for up to 3 years. So again, this is our first year, so we're in a second year now, which we're very excited about. And but it does bother a lot of our workers. You got idle land. Where they come from, rotations are totally different, so they might have better seam clover for a good section of say 3 or 4 months. And so they have because they're growing year round, it's a bit different. So for us, they don't look at it in the same light, but it's sinking in slowly. They see the value of healthy soils after even just a short stint of cover cropping or proper rotations or both. And feeding the soils. The other thing we try to do and we think has yielded a lot of dividends in terms of soil health is feeding our soils with proper amendments. Although we would love to do that more in a more scientific manner, and hopefully Elias and others can help us advance that. We tend to, our basic tillage is pretty simple. We do rely on plastic, unfortunately, but we plow once and then the field generally never sees a plow again. It's a regular moldboard plow. We then use chisel plows and we make our beds, and that's our and then we definitely weed. But that's our basic system. We do have some crops that are not planted in plastic, but they're far and few between. Carrots, and we've even found a variety of carrots that works in plastic. So that's we rely on amendments. We've seen after we started working with a particular company and we started leaf testing in particular, and being very careful and being trying to be as scientific as possible about what we apply to what crop. We've seen, I would think, huge change in the health of the plant. And we hope by extension, to some extent, the health of the soil. But that's one major area that we're working on until we are able to do more like what Jack was talking about, if we can get to that point. And so when Elias approached us about the biome study, we obviously jumped on right away because we know that, understanding how the soil and what lives in the soil, the biome is going to be critical to what we do in the future, and is going to be critical to me handing over my farm, hopefully to my son, and hopefully we stay and our farm stays farming, for forever, if that's possible. That's how we're approaching this, is that we want to hand off the land to the next generation, hopefully in better shape, given the fact that we vegetable farmers tend to disturb the soil pretty much. And I'll stop there. Looks like 15 minutes.
Thank you very much, Zaid. That was great. And I just wanted to say now we have time for questions. We still have another 15 minutes or so. And so if you have a question, type it into the question box and I'll be reading them out loud. The recording of this webinar will be available within a week on the eOrganic YouTube channel. And we'd also be really grateful if you could fill out a follow-up survey that we'll be sending in email later today. So I'm just going to pull up the Q&A box here because I know we had some questions that were coming in. So I think this would be a question for Eli. Can you give examples of the soil pesticides that were used by participating farms?
Yeah, I've been skimming through the comments. I just wanted to go ahead. Can I share our survey tool? Do you mind if I just...
No, I don't mind.
Let me see if I can do this and this. And I'll just show you, this is what the survey tool looked like that the farmers filled out. And so B14 and B15 here were the microbial and non-microbial pesticides that we tracked. Farmers were also able to select an other option or a none option. And then before the presentation, I went through, I had many versions and documented which were the most used in our study. And so spinosad was highly used. Hydrogen dioxide was also highly used. And then regalia or the rutinaria extract was also highly used. So these are just a few, and there's a sprinkling of other soil pesticides that were also used by farmers, but soil pesticides are not highly widely used among the folks that participated in our study, and so I think that's, I mean, our study indicates that if you can at all, reduce your pesticide use from these types of groups, that's beneficial. But it's important for me to say that when we did the study, we grouped all of this soil pesticide use into one variable, whether or not the farm was using a soil pesticide or not. And so we're not really able to identify at this point with our analysis if there are particular pesticides within these groupings that are changing the microbiome in these ways that we think are linked to reductions in a plant's capacity to defend itself. But we do know or have a signal in our data here that there's something going on with the use of soil pesticides, or it's indicating the use of soil pesticides might have a negative impact on the plant's ability to defend itself from other pests. So I hope that's helpful with some clarification there.
Okay, thank you. Let's see, we have another question. Are there any easy ways that farmers can evaluate the diversity of their own soil microbiome?
I read that one too, and I'll just quickly plug for New York farmers, we're working on a rapid soil microbiome decision support tool that we're going to be releasing it eventually. Farmers will be able to send in a soil sample and have it rapidly sequenced within a week or two, and then we'll send them their results. But something like that is not currently available as far as I understand it, especially with kind of the contextual information about what a farmer could do to improve the microbiome. But I did want to go ahead and let Jason or Jack, I think maybe Jack is there and not Jason, jump in, because I know that Stone Barn Center has been working on some microbiome sequencing too, and it might be useful for them to talk a little bit about what they're doing if Jason or Jack is still there.
Yeah, Jason is here. I got to be off video right now, but we are just starting that process. That lay rotation study that we just got a grant for is going to start us on a process of doing that kind of sequencing, but nothing to kind of share as far as like quick ways to do it just yet. We have been working for the last number of years with Yale on quick carbon sampling, and we're actually in a process now to evaluate after, I think it was 5 years, if those quick carbon samples are matching what the longer, more traditional testing methods are showing. So still we should by the end of this year have some results on that. But as far as the other sequencing, still TBD. But I can get back to everybody once I get some answers from Elijah and the rest of our monitoring team.
Okay, and in the study, could you just clarify what kinds of plants were these? All vegetable farms or that you were working with?
Yeah,
Or
these
fruit
were
trees, or were you working with fruit trees as well? If somebody wanted to know. Yeah.
Yeah, for my study, it was all small scale between, very small scale, like a quarter of an acre up to 500 acres of vegetable and small fruit production. So we're talking about berries and things like that. Yep. So there were about 280 farms in the state of New York that fit that criteria, small scale, relatively small scale vegetable production. And then 85 of those farmers participated in the study that I showed results from today.
Okay, great. Okay, we have a question about, it sounds like measuring beta diversity requires the context of surrounding or baseline microbial diversity. Did you use your set of participating farms as the baseline for beta diversity or some other farm set? And do you know how the diversity you found compares with conventional farms in New York?
Sorry, I'll be quick. I did see that comment in there from Sylvie, and we can chat. I know that you're right here in my building. We can chat more about that. But what we did is pairwise comparisons with beta diversity. So every farm is compared to another farm. And then that's a great question about conventional farms. And many people have asked me that question over the last few years about adding conventional farms into the study to be able to compare that to the organic farms. And we just don't have those data yet, but we would love to start integrating some more farms into our network to figure out that question about where organic farms and conventional farms fall in terms of their microbiomes and compare them. Thanks.
Okay, and also Nick wanted to know if there was any collaboration with tribes or traditional methods at the farms.
That's another good question. So the farms that participated in this study, they're just farms that were interested in the having their microbiome sequenced and getting some results about it. And so I'm not able to tell you much more about them. I do believe that some of the farms in the study might have tribal backgrounds, and then we also have different types of farming communities in New York. Some people might call them Amish farming communities, and they go by many different names, and it's important to respect that. And we used a paper survey, and so farmers, I also believe some farmers from that community may have also participated, whereas with an electronic survey, you might have a more difficult time accessing those types of communities. And thank you, and that's a great question.
Okay, here's a question from Joy about whether using a cover crop that reduces pests on one farm could lead to an increase in those pests on a neighboring farm using a different cover, like with some pest migration patterns, or yeah, have any thoughts about that?
It might be for me. I don't
Okay.
I can jump in. I think that's a really interesting question. I think that there isn't clear guidance on that yet. I think that there's a lot of different factors that could influence pest migration. I think ultimately, communication is going to be really important. And I think that a lot of farmers already have that system in place where they're checking in with their neighbors. They're saying, what are you using? Is it helping? Like, what pests do you have on your farm? So I think ultimately there might not be one right answer to that question, but it might be more of a reframing like, okay, yeah, there might be drifting effects from practices that you're using onto your neighboring farms. How do you mutually work on a solution together? So how do you communicate? How do you share practices and knowledge? I think that's an important aspect of that question for sure. But yeah, it's a great one. Thank you.
Okay, we had a question for Zaid. Shelley wanted to know what soil amendments you used.
What soil amendments do we use? Everything from bone meal to compost to some slurry at some of our locations. Our farms are up to 50 miles apart, so every farm might get slightly different treatments depending on what's available locally. We have on our, let's call it on our mature land, we have now gone to a system of very little amendments, other than, N is the primary amendment, where P and K is very adequate in the soils. So it depends. The 50 acres that I spoke about that we just took over. We took it over. It was actually an organic production, but it was a dairy farm, and the P levels were the lowest I've ever seen anywhere. And so that particular farm is getting a lot of amendments right now. And there's where the education part, my field workers think that this was in sod for like 30 years, so they automatically assume that it's going to be like produce based on some experience they've had elsewhere, and it didn't work. So that those fields are getting, a lot of chicken compost, a lot of bone meal, whereas our mature fields are basically mostly getting either we have a formulation that is made up from chicken feathers and some blood meal, small amounts of blood meal, wheat midds, and so on. It's about a 14% N. And so that is used a lot in our high tunnels and not much else. We may use some starter P. We use a fair amount of kelp and fish and kelp meal on everything. And then micronutrients are added based on leaf testing. And we do about 150 soil tests per year. We at the level of even a high tunnel or a bank of high tunnels that have had similar cropping successions over the years. I hope that answers your question.
Great, thank you. Let's see. This is from David. Do you think that the decreased beta diversity of soils for which drip irrigation was used is related to more similar soil moisture among those samples? Was soil moisture measured?
Yeah, I just saw that question in here, David. That's a great question. So when we receive the soil samples from the farmers, we just receive them through the mail. And we unfortunately don't have those additional measures of soil moisture. We're working on getting all the soil properties. We've pulled down some of the soil properties from online databases, but those are very coarse measures. And we just received some more funding to get more of the soil properties, but we just simply don't know. But it would seem as though that the decrease in soil beta diversity, the bacterial soil beta diversity must be related to soil moisture in some way, linked with the drip irrigation and potentially keeping the root area very, very moist and wet, I guess, in that area of interactions where the rise, I guess you could say the rhizosphere, the soil, the roots and the microbiome are interacting. And so yeah, it could be linked with soil moisture, but we just don't have those data. There's just so much data to collect, as you probably know. Thank you.
Okay, we have time for one more question, but I just want to point out on the slide here, if you'd like to find more information about this project, please check out the link that is on the screen about this NIFA ORG-funded microbiome project. And if you didn't have a chance to get your question answered, or if you can think of more after the webinar, please feel free to reach out to the researchers there, and so you can copy this link. right now while we're asking the final question. So here's one from Betsy. Have you considered or tested crop rotations with short-term perennials or sod or winter spring annuals intended for harvest? I'd be interested to hear your perspective of crop rotation to extend plant cover and live roots and their effect on soil health.
Yeah, I mean that if I may, so I mean that's a big thing that we're still grappling with. It's an idea that we've tossed around with again Dan Barber from Blue Hill of crops, because cover crops cost money, and in our case when you don't have the land, what do you do? We've tried some things, but so far there's really, okay, like you grow rye, okay, well then you can harvest, but we haven't really gotten to that point. It's an idea that's firmly in our head, and we need to find, one thing that I might consider in that is fava beans or bell beans, or one of the two. We certainly grow a lot of fava beans, and in my mind, it even, we harvest the pods, and then, some years we get 6 foot tall fava beans, and that all winds up, in the soil. So that is, beans in general, we kind of look to it as doing that. It gives us a crop, and it's also put some N in and put some organic matter. But I would say fava beans might be the crop that we really like for that purpose, if that's answering the question.
Great, thank you. Well, thank you all for answering the questions, and thanks to everyone who submitted questions. And as I mentioned, you will be able to find the recording on the eOrganic YouTube channel within the coming week in case you want to look back at anything that anyone said. And we'd be grateful if you could also fill out the follow-up survey. So thank you so much, Eli, Zoe, Jason, and Jack, and Zaid, and thank you to everyone for joining us today.